North.
Wilmington, and then got a PhD at Duke University, and he retains an affiliation with Duke right now as a research scientist there, and is here now in Santa Cruz as a research affiliation with UC Santa Cruz. So he's living here locally now. And one of the other things that's related to this is he was one of the scientists that used the pointer down in the Antarctic. So I don't know, are you going to talk about any of that?
A little bit.
A couple pictures?
Yep.
But Ari's primary field is looking at foraging and ecological marine animals. He's done a lot of work with large whales, principally putting a variety of different kinds of instruments on animals and then trying to use those to kind of get a better understanding of the foraging ecology of these animals. We don't, Mike Graham and I have been teaching this class for this semester in scientific methods, and we spend a lot of time talking about different methods, oftentimes experiments and all these things where you have a little bit of control over your system. This is an example where you don't have a lot of control over anything other than you getting there with some instrument and then hoping the animals are there, the weather cooperates, and all those sorts of things before you get any data back. So I think Ari's going to be probably giving you some ideas of how these instruments and the kinds of work you might do in this way would give you some perspective on what these animals do. So, Ari, great to have you here.
Thanks. Can you guys hear me okay? All right, good. Well, thank you guys for having me. Thanks, Jim, for the introduction, and Angie for setting this up. I'm also stoked. Like Jim said, I see some of the folks from the Point Sur here, and we had a revolutionary kind of cruise with the Point Sur this past February. The goal of that was to go down and try and put some these recording tags on humpback whales and put out a few satellite tags. Actually, this picture is from the first minke whale that we ever put a suction cup tag on down in the Antarctic. Weather was like this pretty much all the time, right, guys? And we unloaded our arsenal of tags faster than we ever thought we would. We actually had to start contacting cruise ships to get people off of the boat because we had finished our science almost too early. So hopefully we'll get another opportunity to work with those folks. And some of the work I'm going to talk to you about today was from that trip specifically. I don't want to spend a ton of time on this, but obviously the work I'm going to show you is reflective of a lot of different projects, a lot of different sponsors and agencies that were sort of allowed it to happen, as well as the dozens and dozens of colleagues and collaborators that we have that make this possible, from graduate students to folks who help run the ships to program managers. Everybody, without those people, this stuff wouldn't be possible, as you all know. So what I want to do today is sort of introduce you to some of the ecological principles that I use to sort of guide my research. And then I'll talk to you guys about tag technology and the different types of tags that we're using to study cetacean behavior, as well as some of the analytical tools that we're developing, visualization tools to understand what these whales are doing underwater. And I'm going to give you guys a couple of examples of past and current research that we're doing. One is from Stellwagen Bank off of the east coast of the US, off Massachusetts, and then some other stuff about the Antarctic Peninsula. And that should give you guys sort of a flavor for the types of research that myself and my folks are doing. And then I want to sort of put that into a perspective of the ecological insights, but also the management and conservation implications, and looking towards the future and how we can utilize the information we learn a little bit better. So this was sort of my ode to the folks on the Point Sur. This was a beautiful day in Wilhelmina Bay, one of the magical places in the world. And if you're an oceanographer or a marine biologist, this is sort of not typical, well, typical in the Antarctic, but a view where, there's not a lot of stuff in your way. And sometimes you get cases like this. We had a group of about 40 or 50 minke whales utilizing this bay and the sea ice around there, and sort of a really magical couple of days that we had down there. You can see this animal right here. It's got a little instrument on it, and I'll be talking about this tag technology a fair bit later, but this is sort of one of the lucky cases that we get. And as you guys well know, you look out here, you might see a blow here and there, but oftentimes proximity to these animals is pretty challenging. So to sort of start off with a little background, cetaceans, the whales that I'm going to be talking about, specifically the baleen whales, are the biggest animals or the largest animals that have ever lived. And they've got very specific morphological and behavioral adaptations that allow for bulk feeding. This lunge feeding and bulk feeding that they do has allowed them to gain this massive body size and range over large areas of the ocean and basically all parts of the world. And one of the things I'm interested in are the decisions that these animals make and how the distribution and behavior of their prey, different types of prey in different locations, affect their foraging behavior. And then also in terms of the environment that these animals are a part of, how do the physical and biological landscape affect cetacean behavior as well? In that slide you saw before, there was a lot of sea ice. And sea ice is a very important feature of the environment for something like minke whales. For humpback whales, they tend not to be in sea ice, but there's other things in the environment that these animals relate to. Sort of in terms of an ecological mantra, I'm a sort of a firm believer that telemetry is a really incredible tool to both measure and understand cetacean ecology. And a lot of the sort of foundation for that is work that Jim and colleagues did, Don Kroll and folks did in the Wind to Whales project out here in Monterey Bay in the 90s, and really set a good example for how you can actually study whales in a quantitative way and understand them in their environment. And understanding their ecology requires an understanding of their environment very specifically. So everything we do, if we study whales in a vacuum, we're not learning everything we can, and we're not doing justice to the system as a whole. And then the analytical and visualization tools that we have can provide context and convey information in a lot of different ways. And I'm, again, a real firm believer in presenting our information to the scientific community for scrutiny in that sense, but developing curriculum for classes at all levels, from high school to graduate level. And public outreach and education is also a very important part. The stuff that we do needs to impact or be available to as many people as possible, and the visualization tools make a nice display for that. So working with museums and other educational centers is pretty important for what we do. I sort of came up with this concept a couple of weeks ago, talking about sort of the scales that I think of things at, and I think of it sort of in terms of an hourglass. And the thing that sort of is the foundation of the research are looking at broad scale spatiotemporal distribution patterns. And for my dissertation work, I was interested in looking at, in the landscape of the Antarctic, how the distribution of whales related to features of their environment at the broad scale in terms of gyres, ice edges, canyons, those sorts of things. And from there we can build quantitative relationships between cetaceans and their environment. And that allows us to sort of build these models or develop these research questions on fine scale individual-based patterns and use tag-derived information and kinematic patterns of their movement to understand what whales are doing at very fine scales. We can then sort of go back out in scale and look at broad-scale individual movement and behavioral patterns with longer lasting tags like satellite tags. We can then develop individual-based behavioral patterns in relation to their environment. So going from individual feeding behaviors to lunges to days to months of how these animals use their environment, and eventually generate predictive models for relating environmental change or human impacts on cetacean ecology. And I'll sort of end with some of the human impact things that I'm interested in. So in terms of foraging theory specifically, if you're a good predator, there's a couple of things that you have to keep in mind, and the first are your physical constraints, right? So this puffin over here, the morphology of its beak is the limitation here, not the brain saying I can fit as many of these fish as possible into my mouth, but there's constraints there on how many prey that animal can actually handle at one time. The physiology, how deep you can dive, how long you can hold your breath, and all those sorts of things, as well as your sensory capabilities and how you're able to gauge your environment and find prey, are all things as a predator you need to be aware of, or are conditions. Well, secondarily, prey, also to give them a fair shake, prey behave as well. And this pufferfish is a good example of something saying, please don't eat me. I don't want to be prey. And so predators have to sort of weigh the balances of what you can do versus what your prey is going to allow you to do. And because of that, the behaviors that lead to prey capture are going to vary quite a bit in the timing of when you feed, whether it's crepuscular or diurnal, and how you go about doing that, the duration or how long it takes you to feed and get all the energy you need, as well as the constitution, whether you're a gulp feeder, whether you take small bites, whether you feed cooperatively. And all of those things, or this ability to be plastic and have sort of flexibility in these appetitive behaviors, is what makes a really good predator. And I can give an example that I think folks will probably recognize. So we'll talk about beer as being a prey. In the case here, beer is acting as a single individual unit. We've got people here taking advantage of prey in that sense. It can also aggregate a little bit further into the mini keg. So think of this as a smaller school of fish. And then obviously the ultimate is not the beer truck, but a full keg. So you've gone from an individual prey to an aggregated set of prey. And if you're a good predator, you can take advantage of this prey when it's behaving differently. So I get to throw my friends under the bus a little bit. And Jim and I have a good friend named Charles, who's a researcher in Hawaii. He runs the monk seal foraging ecology program. That's Charles right there. This is Charles and a couple of his colleagues taking advantage of beer when it's acting sort of in its singular form. And when Charles gets off a research boat after 2 months, he's hungry or thirsty and is able to take advantage of prey when it's acting in a slightly different form. And while this isn't Charles, this is still a pretty creative way to take advantage of prey, right? So a keg is clearly not designed for one person to take advantage of, but if you've got plasticity in your foraging behaviors like this guy from the University of Virginia, I think, you're able to do that. And this is some pretty creative behavior going on. So that's from bestkegstand.com, I believe is the website. So you guys can add to that if you feel it's necessary. So to sort of put a little context to what we know about whales at this point and their behavior, blue whales have been one of the most well-studied animals in this respect right here in Monterey Bay. And it's been found that they perform shorter dives at shallower depths. Rather than going down for 20 minutes and feeding at a shallow depth, it's more efficient for these animals to go down, feed once, come back up to the surface. And their foraging time increases with increased travel time. So the deeper an animal goes, the longer it takes you to get to your prey, the longer you should stay down there to take advantage of it because of the energetic consequences of traveling. And these shallow, short dives that animals do actually increases their feeding rates. So for a blue whale, it takes a massive mouthful of water that's almost equal to its body mass. It takes a long time to process that prey. And so it actually is quicker for the animal to come to the surface and process that prey and go back down than it is to stay at depth and do that. Humpback whales, on the other hand, sort of the other species we know a fair bit about, they've been observed foraging shallower than the maximum prey density. And this is something that sort of puts into context how these animals feed optimally and what are the energetic costs versus the gains that they get and how they locate their prey. They've also been shown to be selective about their prey species. I'm going to talk about humpbacks from Stellwagen Bank in the Antarctic. In Stellwagen Bank, they feed on sand lance, herring, and krill, depending on what's available for them. The Antarctic, there's really only one prey item, and that's krill, but they're able to be flexible in what they take advantage of. In a different type of animal, in diving birds, where you have the ability to do a bit more sort of experimental work just because you can handle the animals a little bit better, like Jim was mentioning, it's been actually shown that they increase their dive depth with increased prey density. And this is a really important aspect of diving and behavioral ecology that I'm interested in trying to understand with whales. And it's logistically a bit difficult to do. So we can think of whales or cetaceans as central place foragers, or any air-breathing animal as central place foragers, because you have to return to the surface between every single dive. So the animals are not sort of at their whim to take advantage of the environment. They have to weigh the costs of traveling to and from that central place every time they go and feed. Hey, I'm giving a lecture now. Look at that. Awesome. Thanks for reminding me 20 minutes in, right? Yeah. And so if you think about prey depth or where the prey is in its availability of these animals, as the prey depth increases, as I mentioned, so do the travel costs associated with accessing that prey, and thus your energy gain should increase. So the deeper you dive, you have to make it worth it for you to dive deeper, so you should be taking in more food at that point. So you're going to increase the time you spend foraging at depth relative to a shallower dive. But this is all assuming that prey is at a constant density and that there's no gradient in how prey is distributed in the water column. So we're interested in asking questions about whether or not this is sort of a real situation or how whales take advantage of that. So optimal foraging theory, this is my friend Brandon Southall up there. He's at Santa Cruz as well. That's eating sushi in La Jolla. In terms of optimal foraging theory, or how animals optimize their behavior, it's been postulated that species have evolved these strategies to search for and maximize net energy gain with respect to the costs of acquiring that energy. And for diving animals, understanding their time-energy budgets requires a knowledge of the mass of the animal, the metabolic rate of the animal, as well as characteristics of their prey. And for things like cetaceans, where measuring a field metabolic rate is a very difficult thing, the next best thing we can do is start to understand how changes in the prey, the density of the prey, the constitution, how big prey patches are, the depth that it's found at. Understanding how that affects the animal is sort of the next best thing that we can do. So the objectives of sort of my research at a broad scale are to quantify daily activity patterns of humpback whales in relation to prey and test these predictions of optimal foraging theory. And the thing that this is going to do is it's going to give us some really good information on the species sort of in its own vacuum, but it also gives us some information on the ecological interactions between predators and their prey that can be looked at across systems and across species. And then it also allows us to better understand how changing environments are going to affect these animals and systems in general. So we've all, sort of everybody in the room has made the decision that studying land animals is probably just too easy, right? You can go to Africa and sit in a Jeep and watch a leopard walk around and hunt and take down the impala and then eat it right in front of you, and there's no guesswork, right? You have to do a model, you don't have to do any statistics, you can just kind of watch it happen. And we've all decided that that's not good enough. So we've decided to study something that doesn't occur very frequently at the surface and doesn't give us really an opportunity to see what they're doing. So the next best thing is to make friends with some really smart engineers and come up with ways to measure what the animals are doing underwater. And this is that minke whale again, and this is wearing a tag called an AccuSound, which is developed by a guy named Bill Burgess who lives just up the road in Felton. And it's a multi-sensor tag that attaches to these animals using suction cups. And it's one of the primary tools that we've started to use to measure the very fine-scale movements of animals in their environment. Another tag that we use is called the D-tag. And this is an older version of the D-tag here. Essentially the same kinds of tags. They're non-invasive, so they really don't affect the behavior of the animal that you put them on. They attach with suction cups, just straight up silicone suction cups. They have a duration of about 24 hours on the animals, and depending on the species, some last longer than others. But they've got a bunch of sensors on board that measure the time, the depth, temperature, as well as acceleration and magnetometers that work in 3 dimensions. And anybody with a smartphone, it's literally the same sensors you have. I downloaded a nerdy little app on my iPhone that shows you the magnetometers, the gyros, and the accelerometers. So you basically have a D-tag that's just not pressure rated every time you've got your little phone out. What's cool about these tags is that they sample at 50 hertz. So you've got some very, very fine scale measurements of behavior. It also records audio up to 96 kilohertz in one tag, up to 192 in another. So we're getting information on what the animal's acoustic environment is like, as well as the sounds that the animals are producing, and linking those behaviors to acoustics is another interesting facet. They have flash memory, just like a digital camera does. You pick them up at the end of the day and you plug them into your computer and offload the data. And they've got a standard VHF system in there so you can keep track of the animal while the tag is on them as well. Deploying the tags is obviously the most fun part of what we do, so I figured I'd show you guys some good videos. This is putting a tag on a blue whale off coast of Southern California. This is work we're doing with John Kalmakidis from Cascadia Research Collective. You'll see the flukes of the whale sort of appear right over here. Blue whales are big, and you take advantage of them when they come up to breathe. Once they're up, they don't have a choice, so you can kind of rush up on them and put the tag wherever you want. See, the tag looks pretty minor in comparison to the body size of that animal. First time I tagged, there's John, he's a crazy dude. First time I tagged with him, I wasn't really prepared for actually getting on a plane when you're approaching a whale. That's kind of opposite of what you think you should be doing, but it's obviously an effective thing. This is putting on something called a critter cam. I'll talk a little bit about this. It's a video recording tag as well, made by the National Geographic Society. They threw a red herring at us over there. This is a bubble net being produced by animals in Cape Cod. You'll see about 5 or 6 whales coming up. Humpbacks kind of give themselves away. They tell you where they're going to be, which is, in the whale world, pretty unusual. And then these animals all have a big mouthful of water right here, so they've got to process that and push that out through their baleen plates before they can do anything else. So we take advantage of that. And I usually turn the audio on this one down because that tag is kind of heavy at the end of the pole, and I start swearing at the whale and telling it to breathe, please. And it eventually does, thankfully. And then the tag goes on. The critter cam has just one suction cup, and it also records animal-borne video. Gives you a really interesting perspective of what the animals are doing and how their behavior relates to things around them. You can tell the behavior of those animals wasn't really affected by putting the tags on. And then in the horrible and difficult to work in Antarctic, these are the horrible conditions the folks on the Point Sur and I had to work in. These are a couple of humpback whales and a Beaufort Zero. and you'll see the effect of tagging has on these animals. This animal's sleeping and remains sleeping when the tag goes on. Eventually it sees us, and he just woke up for about 2 seconds. About 20 seconds later, the whale is back asleep. And there's that little orange tag, that's the AccuSound, right on the back of the whale right there. So it's of course we only show you the ones that work, right? So it's a simple procedure, it always works. It's always flat, calm too. But that's when we interact with the whale, and then it's let the whale go and do its thing and figure out what the environment looks like around it. When we get the tag backs, though, we've got to figure out and make sense of the data. And so one of the things we've done is work with a guy named Colin Ware at the University of New Hampshire, and he developed sort of a volumetric movement software package called Fledermaus a bunch of years ago, and we worked with him to sort of turn that into a package that would allow us to look at tag data in 3 dimensions. Instead of just an XY plot in 2 dimensions, we were interested in how these animals are using their environment around them. So what this package does is it turns the track of the whale into a ribbon, and the ribbon relates to the orientation of the whale in space and shows you when the animal is going up and going down. The other thing that's really nice about it is you see these blue and red little shark fins. And what this is the acceleration signal away from the central axis of the whale. So that indicates when the animal is fluking. So right here we can tell something really interesting about the whales, that when this animal dives here, it sinks to the bottom, it's kicking quite a bit at the bottom, and then it has to kick on the way to get back to the surface. So the simple thing that we can learn from this is that this whale is negatively buoyant. So it's saving energy when it dives, but it's got to use up energy when it's coming back up to the surface. So we can start to use sort of simple metrics just from visualizing what the whales are doing to understand their behavior. Well, the next thing we want to do is figure out when these animals are actually feeding underwater, and what's the signature in the dive record that allows us to see that. And so one of the things that we know is that when a whale opens its mouth underwater, if it's swimming, what's going to happen to the speed of the whale? Right, it's going to slow down. It's essentially opening up a parachute underwater, so the acceleration of the animal is going to slow down dramatically. Well, we don't have speed sensors on these tags, but the next best thing that we have are those are the hydrophones. And so those are measuring flow noise that the animal is giving off as it moves. So on the bottom, what you see here is actually the flow noise of the tag. So imagine you're at a stoplight, you roll down the window, there's not a lot of noise. As soon as you start to accelerate, a lot of noise starts passing by. So we correlate that with the acceleration signal of the tags, and we can generate an estimate of the speed of the whale. So what we look for in the tag record are places where the speed of the whale increases and then decreases dramatically when it opens its mouth. And this, we would presume, is when the animal is feeding. So in this case, where you see these red and blue fluke strokes is relating to right here, and then right at the end of it, right here, is where that animal opens its mouth and lunges underwater and takes a bite. So you can automate this, or you can give it to your graduate students to try and automate and come up with a filter to look for these sort of dramatic changes in the acceleration of the animals. And then you look and you can see in the dive record, they start to sort of appear all over the place. You see one here, here. So you can then go back through your tag record, pick out the time, pick out the depth of when all these things happen, and that's your metric of the feeding rates of these animals. So this was a method that a good friend of ours, Jeremy Goldbogen, sort of came up with about 8 or 10 years ago, and we've started to sort of formalize ways to do this and use other sensors to come up with the same information. This is another sort of set of the same sensor signals from an article that Jeremy and I published in Bioscience recently. This is the fluking acceleration of the animal here. This is the simple XY plot of a minke whale here. And on the bottom, what you have here is called the normalized, it's called the jerk. A graduate student in Norway came up with this concept where you basically take all of the acceleration, put it all in one sort of big package, and look for changes in that total acceleration to indicate when the animals are in a dynamic position, when their body is moving or dynamic acceleration is changing. So the peaks in this dynamic acceleration, or the total acceleration of the animal, relates to when feeding events occur. So in this animal, this is a minke whale that's actually diving to about 40 meters and lunges 24 times in one dive, which is pretty extraordinary for an animal. A blue whale will max out at about 4 lunges per dive. But a minke whale here, you can see all these peaks in this dive record on this one dive are when that animal was feeding. So now to sort of show you where we did this work, we'll take you to the western Antarctic Peninsula. The area right here is the Gerlache Strait. Andrews Island, where Palmer Station is right down here. And this is sort of our main testing ground for a lot of the understanding how whales forage in relation to their environment. I'll show you sort of a little cartoon that goes through the methodology that we use, and the thing that I haven't talked about at this point is measuring prey. So we're zooming in on a place. Captain Rick, what's this one right here? You know this one. This is Wilhelmina Bay right here. Yeah, yeah. So this is Wilhelmina Bay. This is turned into sort of like our little temple of whale foraging behavior. So what you see here is a really cool way to visualize data. So we're now out in a little Zodiac. We've got a 38 and 120 kilohertz echo sounder going. We're trying to track a whale that's tagged. So the white spaghetti in the background is a whale track. This is us in the small boat. Behind us in the big boat with a 4-beam, actually that's an 80CP, but it's essentially a 4-beam echo sounder as well. It's mapping the prey around where this whale is. So you can see that there's not a lot of prey in the upper parts of the water column, maybe a little patch here, but as soon as you get down in depth, and you'll see it sort of come into the picture, you get a massive layer of krill, starting in this case at about 150 meters down to about 400 meters. So what I want to show you from here is A, what we do, B, how difficult it is to actually map prey quantitatively around where an animal is, because the beam of these echo sounders is about 7 degrees. So it's essentially just like shining a flashlight through the water. And if you wanted to see what this entire room looked like, it would take you quite a while just mowing the lawn back and forth and doing that. So what we try and do is essentially that, is stick around a tagged whale, map the prey as much as we possibly can by approaching the whale, going past it, and making these sort of cloverleaf patterns. You can sort of see them right here around where this whale was foraging. And that gives us an idea about what the prey is like locally, and then as you sort of radially move away from the animal, what the prey is like in the environment. So it's nighttime, the big boat picked us up there. The red is going to be the track of the big boat, and so in May in the Antarctic at night, you've got about 18 hours to go blind. And so what we're doing here is basically using that VHF signal to tell us where the whale is in relation to us, and then we're just sort of going around it and mapping prey in an area and hoping that the prey that we measure is a place where the whale is going to coincidentally sort of go through. So here's our humpback whale. It's taking breaths at the surface. It's going down to feed now, and you'll sort of see these little kind of roller coasters are where the whale is feeding. There's a lunge, there's a lunge. It's lunging its way up to the surface, kind of rolls around a little bit. It's happy. It's going to go back down and start lunging again. So you can sort of see where each of these feeding events occurs by this whale. And like I said, if you're lucky, you'll find out that the animal lunged in a place where you've measured the density or the distribution of prey coincident to where that whale is. So it's not a trivial thing, or it would have been done a lot recently. So when we get the tag data back, I mentioned sort of these two-dimensional dive profiles are the very old school way of looking at these, but they're still very useful. And what this shows you is 24 hours in the life of a humpback whale in the Antarctic in May. And there's a few things that kind of stick out. So on the bottom axis, you have time. You have depth on the y-axis here. So this is down to about 350 meters. The thing that you see here is rest, right? So every time where there's this punctuated gap of nothing and then a deep dive, this is the whale actually sleeping. So that, showing you guys putting that tag on the whale, those whales are out cold. They're just hanging out at the surface, doing nothing, resting, literally resting. Then they do these deep exploratory dives. And what we think is happening, you saw how deep that prey was that we were mapping before. I think what happens is these whales fall asleep for a couple hours, and then they're going to dive deep down and be like, okay, just making sure I'm here, go back to bed, keep doing that, and make sure that prey, that A, they're above prey, but also that it's too deep for them to efficiently forage in, because these dives are between 250 and 350 meters. And for a baleen whale, that's pretty darn far. But then you see this initiation of feeding that starts at about, on this graph, at 2 p.m. And again, in May in the Antarctic, that's when it starts to get dark. And then the depth of that feeding by this whale continually gets shallower and shallower as krill vertically migrate, and then it goes back down later at night. And this is sort of as robotic a whale as we have. So this is just a representation. Not all whales do this, but we show this one because it shows the trend very nicely of them starting to feed in one manner, tracking that prey through the water column as it moves up and it moves down in the water column. So what we want to learn is, do the feeding rates change as this prey moves up and down? What's the density like of this prey as it's moving in the water column? And how does that affect what these whales are doing? So if we go through 10 or 12 animals and we pull out every single dive and categorize it as either being a traveling dive, which is essentially a dive where there's no feeding, just resting at the surface, a feeding dive, or one of these exploratory dives, we see that animals travel, they rest throughout the day, so going from midnight to midnight. The thing that stands out, though, is that you have this massive gap in here where the whales literally do not feed. And that's from about 9 in the morning to about 4 in the afternoon. The animals literally are not feeding at all. Then they feed completely consistently as soon as it gets to be afternoon and into the nighttime, and then into the next morning. So there's something about what the prey are doing that means that is affecting how these whales decide to forage. So if you look at the foraging depth of these whales now by hour, so this is sort of aggregated for about 15 whales. When they start feeding in the early afternoon, it can be very deep, but it can be also very variable. And then as it gets towards nighttime, like you saw with that one whale, the animals start feeding up to about only 50 meters deep. And then as you get throughout the night, it starts to get a little deeper, and then as soon as it drops off the next morning, the animals stop feeding. So, with respect to what they're doing at depth, we found this really good relationship between lunges, the number of lunges, and the deeper that these animals dive. So the number of lunges per dive increases with dive depth. So for the animals that are diving between 0 and 25 meters, they typically only lunged one time. So these yellows are lower numbers, going up into the blues and purples are higher numbers. And as you increase in depth, you see that you increase the frequency that you have a lot of different lunges, or a higher number of lunges. This is a pattern that we see in blue whales. I mentioned blue whales taking one lunge shallow and then coming up to the surface to breathe. When they feed deep, they lunge 4 times. And this is something that directly supports optimal foraging theory, that the animals are taking advantage of that time it takes for them to go down deep. They're lunging to maximize that time down deep. The next question is the food any better where it's deep? Otherwise, why would you dive at all? So let's look at what we find out about prey behavior. So this is, again, going from sort of midnight to midnight, and these are just several different measures of krill patch dynamics in Wilhelmina Bay. So the depth of the prey patches, you can see at midnight is very shallow, and then it decreases or it gets deeper during the day. When you look at the patch height, this is another interesting feature. During the nighttime, the patches are pretty dispersed. They're pretty thick in the water column. During the day, they become much more aggregated. And then at night, again, they become higher, sort of more broad. In terms of the density, the SV, you see that the patches are less dense during the night, and they're more dense during the day. That makes sense. If it's the same amount of krill taking up a smaller space, the krill are actually going to be denser. And then this is just sort of a measure below that of how much area in the water column krill are taking up. It's basically the same thing as patch height. But during the nighttime, krill are taking up a huge portion of the water column. During the day, they're taking up a very small portion of the water column. When we relate this to what whales are doing, we find that humpback whales in the Antarctic at this point are feeding when the krill are less dense. So the no versus yes are the hours when the whales are feeding versus not feeding. So the krill are denser when the whales are not feeding, which is kind of counterintuitive. But the prey is much more shallow when the animals feed, and they're in larger patches when the animals feed. So that tells us something about what the animals are cueing on specifically to help them find prey, or to find enough prey. We were able to find a really, really good relationship in predicting the depth that a whale was going to feed at when we took the measures of krill patch density as well as depth and area, and sort of put these into a linear regression model. And we get a really high R-squared value here. So there's a really good relationship between these variables, some combination of density, depth, and area that affects what the whales do. And then the most sort of interesting and most difficult thing to do was to measure the density of krill at the locations where the animals were lunging or doing these actual feeding events. And for all the animals that we tagged, we only have about 300 data points in here. So there are only about 300 lunges where we have the density of krill within a couple hundred meters and within a minute or two of the data being collected. But what we see, regardless of the year, is that the deeper the animals go, the denser the krill are that they're targeting. So the krill we know are not distributed evenly in the water column. The density is not distributed evenly. And these whales are taking advantage of the krill. So the deeper that they go, they have to feed more. And the krill are even denser. So it's more effective or more efficient for these animals to go deep and feed on denser krill patches. So I never get to this slide before having talked about all of this stuff. So it does bring up the question, like I mentioned before, it's like, why would you dive at all? If prey density didn't change as krill migrated vertically, why would you ever feed deep, and why not just wait for these animals the krill to come up to the surface. Well, we showed that as you dive deeper, you're actually targeting denser prey. So there's some relationship there that makes that a useful strategy. Our data do suggest that deep krill are also dense krill, and that it's sufficient for whales to make dives to that depth to target prey in dense prey patches. And this brings up sort of a point that Jeremy and I raised in a different paper, that the efficiency of lunge feeding increases with increased prey density, and that makes sense. The more food that's around you, the more you can get into one mouthful. The confounding thing for us that we found in the Antarctic, though, is that those patches tend to be distributed much more patchily. So these whales are weighing the cost of, do I feed in an environment where there's a lot of prey dispersed around and I don't have to have any search time, versus trying to find a patch that might be harder to find, but that might be a little better to feed on. So an increase in prey density targeted will decrease the number of lunges that's required to achieve your daily energetic needs. And this is really important because we figure that humpback whales are these awesomely effective feeding machines. In that two-dimensional plot, you saw the animal was lunging or feeding for about 16 hours straight. So in order to fill its energetic demands for a day, it had to feed for that long. So either they're taking in an order of magnitude more than we think, which pretty much isn't the case, or lunge feeding in this environment isn't that great. They're never getting the mother load. We've never seen a whale feed for like 20 minutes and then boom, take one lunge and then just stop. They never get a ton of krill in their mouth at one time. So whatever method they're doing that they're using works, but it never works much better, from one lunge to another, which is something that's interesting, even for a really good predator. So putting this in the context of the Antarctic, if we dig a little deeper, sea ice, which is sort of an integral part of the environment in the Antarctic, the duration and the extent of sea ice in the Antarctic Peninsula is decreasing. This is going to have some consequences that hopefully we can start to look at and hopefully take down the point, sir, and start looking at this in a little more detail. You increase the availability of prey to things that are ice intolerant, like humpback whales. So knowing how minke whales that feed in the ice take advantage of krill versus how humpback whales take advantage of krill in open water, it's going to show us that humpback whales feed a lot more than minke whales do. You also decrease the availability of prey to these pagophilic species like Adelie penguins and minke whales, and this is going to change the balance of competitive interactions in that environment, and you're either going to displace animals that need sea ice to live, and you're going to decrease the amount of habitat, you're going to decrease the carrying capacity of that environment for those animals. So we're sort of at the cusp of seeing some very dramatic changes in the ecology of this environment, that the more we learn ahead of time, the more we'll be able to understand how these changes are actually taking place. So in summary, for the Antarctic, as I mentioned in fall, these whales prefer shallow, larger, less dense krill patches. Within those patches, though, they feed on dense prey. So the deeper they're feeding within that patch, the denser the prey are. And I think that one of the reasons this is so is because when I showed you that animal sleeping at the surface or that resting behavior, these animals are actually positively buoyant at the end of their feeding season. They look like potatoes with flukes. They're these big fat things in the water. So they're limiting their energetic demands of diving deeper by waiting for those krill to ascend in the water column and then just feeding consistently in very shallow water. And these all support these optimal foraging theory models. So to sort of switch gears and move to Stellwagen Bank off the coast of Massachusetts, for the non-native New Englanders in here, Fenway Park is right over here, I think. This is Stellwagen Bank National Marine Sanctuary, and this is Cape Cod right over here. This is a very urban marine sanctuary. Its proximity to a lot of human activity makes it a unique place to work. There's been long-term humpback whale research going on there since the 1970s. The initial animals where photo ID or using flukes to identify individual whales was pioneered there. And it's sort of this nexus of industry in terms of shipping as a port, recreation for fishing, for science, conservation, and management through the National Marine Sanctuary office there. And it's a really cool place to work. And in this area, humpback whales do a lot of feeding at the surface. It's a very dynamic environment, like you saw when we were putting that tag on. And the thing that they're feeding on mostly are sand lance. The ichthyofauna in this area used to be dominated by herring about 25 or 30 years ago, but it has sort of changed rather quickly, and sand lance is now the dominant prey species, and it's really affecting how these animals feed and the behaviors that they do. So one of the things that these animals do is bubble net feeding, and what you see here is sort of an aerial view from a blimp of an animal that's made a bubble net like this, sort of in making smaller and smaller bubbles and then sort of feeding by itself. And what you see here are track plot images of an individual whale sort of over different periods of time. So this is looking down on the animal. it starts out down here, it makes a loop here, another loop, and then comes straight up to the surface through here. And then on the upper right, you see that animal doing it a few times, and then below that, you see it doing it even more and more times. And the thing that this really speaks to me is that you see a pattern that's repeated over and over again. It's not this random going around and lunging, but you see a repeated pattern that this individual whale is doing. And if we animate that, this is one particular whale. I can't remember the name of this whale, but it's doing something that's very distinctive. And some colleagues of ours just published a paper in Nature about this, what's called lobtail feeding or kick feeding in these animals, and that it's able to transmit this behavior through a population of animals in a way that really indicates that the animals are social and learning and have a lot of ability to interact and learn from each other. So what you see here is the animal is diving down. Where the yellow is where the animal's rolled on its side. So this animal rolls on her side. She'll start blowing bubbles right here. She's going to make a ring of bubbles, come up to the surface, and then you'll see like this little seesaw action right there. So she's kicking the surface with her flukes. Then she goes back down and then comes right back up into that spot where she kicked. And this is a very sort of prototypical sort of feeding behavior that we see whales in Stellwagen Bank do. What's really cool about that area, we've been working there for about 10 years. We've deployed about 150 tags now. You start to see individual patterns, and you see characteristics that are really, really interesting. So this animal does a loop all at the same depth and comes up. This animal probably kind of gets dizzy. It's doing 3 sort of loops as it comes up in the water column. This is that same animal you just saw doing 2 loops. I don't know what this guy's doing. This is sort of a confused animal or a younger animal, but what you see here is an amazing amount of individual variability in what these whales are doing. So the end result is the same, an animal at the surface with its mouth open, but the constitution of how these animals do it is very individualized and very stereotyped across animals, which is something that we didn't understand until we could really look at the dive profiles in 3 dimensions. The other thing that we found that was really interesting is what was making these animals do this? Well, we found if you look at prey patches, there's vertically oriented prey patches and horizontally oriented patches, and these are all sand lance. What we found was that humpback whales will target these vertical prey patches in the water column significantly more frequently than these prey patches that are horizontally oriented. And I sort of call that the Pringle can hypothesis, that if you've got a table and all your potato chips are spread out and you just stick it over that, you're just going to get a thin little bit of Pringle. But if they're all stacked up, you can put the can over the entire thing at once and scoop it up and you've got everything. So the whales are actually locating these patches that are vertically oriented and bubble netting around them. And we see a very similar pattern in Alaska where the animals are feeding on herring, and they'll bypass these large patches of herring that are very dense but that are bottom associated for these patches that have these chimneys of herring that are sort of dispersed in the water column. So they're pretty savvy. The other thing that we found out, and which was completely serendipitous, was if you leave a tag on a whale overnight, you learn some really cool stuff as well. What we found is that these animals are actually feeding on the bottom at night. There are some researchers in the 90s that thought there's all this scuffing on the side of humpback whale jaws in Stellwagen, and they thought the animals are probably going down and taking advantage of their prey at night, but there was no evidence of that, and we got lucky and had a couple tags stay on overnight, and we saw this really interesting behavior where the animals will go down to the bottom. That yellow, again, is where the animals are just rolling onto their side. These animals are basically going down, rolling on their side, upright, rolling on their side, basically mowing the lawn back and forth over and over and over again. And we think that this is evidence of these animals bottom feeding. So what's really cool, again, is the more tags you put out, the more you see some really cool individual variation in this. So here's that same animal going in a line and sticking its nose in the mud. Here's an animal that's sort of working itself back and looping through the same area over and over again. Here's an animal that's basically making circles in the mud as it rolls on its side. And then D is, I think, just very similar to A. But again, some very cool individual variation in how these animals are taking advantage of their prey when it's behaving very differently. The other thing that we noticed, and this is where I'll sort of bring that critter cam stuff in, is if you're looking at one whale in a vacuum but you know it's part of a pair, you might be missing out on something. So we tagged these 2 whales, Echo and Tectonic. We know that they've been feeding together in the summertime for about 5 years. When we looked at their dive profiles and we looked at the roll that the animals were making, as well as their actual heading and then the pitch of the animals, you see this really kind of cool pattern where the animals were rolling opposing each other and then coming back together and rolling opposing each other. So this is a still frame from the critter cam What you're looking at is a critter cam that's on the back of the whale on the right, and this is its nose right here. This is its right pectoral flipper, so it's rolled over on its left side. And this is the back of a second whale. So what these whales are doing is actually going down and they're rolling back to back with each other, and that's what you see sort of right here. The whales are rolled over basically back to back on each other, heading the same direction right here. So we think this is a cooperative feeding strategy by two animals. What they'll do is they'll go down, and if I had time, I'd show you the video. They basically go down, roll on their sides, bonk into each other a little bit, and they fill up. You'll see their mouths distend full of prey. And they do this over and over and over again. So we had no idea that A, bottom feeding existed, B, that it possibly could be a coordinated type of feeding behavior as well. So it's probably likely that animals that spend a lot of time with each other are able to coordinate their behaviors and know each other well enough that this is a strategy these animals can take advantage of. So what's really cool about this, though, is it also is a great example of how humpbacks are taking advantage of changes in their prey. So what's really well known is that sand lance during the day are found throughout the water column, but at night, sand lance go down into the substrate and burrow into the substrate, and that's where they spend their nighttime. When we look at the frequency of surface feeding versus bottom feeding, we see that at nighttime, and here's sunrise, you have no surface feeding or bubble net feeding, and it spikes during the day, and then it decreases. It sort of ends right at sunset. If you overlay this with the frequency of when bottom feeding occurs, it's a complete sort of opposite. So this is nighttime, the animals are bottom feeding. Right as soon as sunlight hits, the whales change their distribution in the water column. The animals don't bottom feed, and then when the prey start to move down into the bottom, they pick this up again. And one way that we were able to understand that this was relative to prey was when we looked at the maximum prey density throughout the entire water column. It actually occurred about 3 o'clock in the morning or 4 o'clock in the morning. This is when the sand lance started to change their behavior from being at the surface to being down in the substrate. So some really interesting patterns in how they take advantage of their prey. So in terms of Stellwagen Bank, what we've found so far is that there's very pronounced dye yield differences between surface and bottom feeding, and these are coincident with available light. So surface feeding is a daytime activity. It probably involves a lot of coordination between animals, and it's probably a very visual way of feeding. When you look at the critter cam data and you look at the videos, a lot of times you'll see whales rolling on their side, and you can see a backlit prey patch right at the surface that they're taking advantage of. We see some really cool individual variability in cooperative feeding behaviors, both at the surface as well as on the bottom. And by combining traditional fisheries acoustics, the information we get from the echo sounders, with novel tag and visualization technology, we've got some really remarkable ecological insights that not only are useful here, but that you can take and you can apply in other systems and other species in different environments. Sort of looking forward, and the things that really excite me, sort of combining telemetry and analytical and visualization techniques, is to understand the kinematic patterns of these behaviors, right? So we can look at these tag records and know exactly what the animals are doing just based on the different orientations of their bodies. It allows us to understand these ecological interactions when the animals are taking advantage of their prey, and it also provides this foundation for expanding the scope of our work. So we can start looking at more broad spatio and temporal scales. We can look at interspecific relationships if we can tag blue whales and fin whales in the same environment at the same time. We can understand cetacean in behavior in relation to environment and human impacts, and it provides a template for comparing species in ecosystems. So moving this kind of work and, again, starting to revamp what was done in the Wind to Whales project out here is really exciting for me to be able to sort of move ahead with that. But then there's also this question of so what? We talk about whales and their environment and their ecology, and sort of what are the so whats? And I'm going to give you sort of two things that are useful for me. So what you see here are two animals that are tagged together. This is a mom and a calf in the Antarctic that we put D-tags on together. And this is the first indication or the first example of seeing how animals might possibly learn how they feed and what they learn from their mothers and how they mimic some of these behaviors. So on the right you have mom in the yellow track. The calf is in blue. You see that positively buoyant calf just kind of shot up to the surface there. Kind of wiggles around at the surface. It's not a real good swimmer. Takes a lot of energy for that calf to swim down. You see it fluking all the way down. But what it's doing is essentially mimicking everything that mom does. Goes down to the same depth and follows right behind her. So this is both spatially and temporally accurate. So this is a really good indication that later in the year when mom is feeding, that calf's going to go down with her, see what she does, and basically mimic what she's doing. And that's probably how this calf learned how to feed, which is pretty cool. And we don't get opportunities to tag multiple animals like this all the time. So this was a very cool way to show how teaching or learning might occur in cetaceans. The other thing is sort of understanding human impacts and putting this in a conservation framework. One of the things that we're really interested in doing in Stellwagen Bank, and that you can apply to the Monterey Bay, to the Channel Islands, the other marine sanctuaries, is how human use affects these animals. So we know that animals end up on the front of some ships. They get struck by ships, either by propellers or the bows of boats. They get entangled by fishing lines. And basically what we're trying to do is on the right there is show we understand now how these whales are using different parts of the water column. We know at different times of the day they're behaving differently. We know at night what they do during the day. And we can start to put together a picture of where these animals are at the most likelihood of being either struck by ships or getting entangled in fishing gear. So we can make recommendations if we know the substrate type that the animals bottom feed on, or the places where they bubble net feed, and make recommendations and say, look, in Stellwagen Bank, you've got these real hot spots of where whales are. This is the shipping lane as it used to occur in that dotted line, or excuse me, on that solid line. If you move that to where that dotted line is, you can remove yourself from a lot of potential interaction with these animals just by showing spatially where these animals do their business. And so applying these kinds of metrics that we learn about the behavior of these animals to what we do as humans can be pretty powerful. And then obviously entanglement and fishing gear is sort of the other one that happens more in the water column. So just kind of to summarize, humpback whales specifically have unique adaptations that allow these really extraordinary feeding behaviors. And by combining tag data, prey mapping, and visualization technology, we have a much better understanding of humpback whale foraging behavior, as well as relationships between whales and their environment and other predators. And this can help to lead to more successful protection, conservation, and management of the species. I think that's all I had for you guys. I appreciate the time, and I'll be happy to answer any questions. Yeah. Yeah, a good question. If I'd shown the video, would have seen the sand lance were actually a complete carpet on the bottom. They weren't in the substrate. They were right at the interface there, and it was just a mat of sand lance. So they were going down to the bottom, and when they open up their mouths, it's a little bit of a vacuum. They're just sucking up prey right off of the substrate. The animals that are sticking their nose in the mud are actually scaring the fish out of the substrate, and then when they right themselves, they open their mouth. Yep. Yes, sir.
You often hear about how much prey whales could be taking in. You talk about hitting the mother whale with that feeding lunge. I didn't really see that number. Do you think that's a physiological thing, where maybe their stomach itself isn't capable of holding their maximum catch from their mouth, or maybe they're just able to just pen down there, they're just not doing it?
Yeah, that's a great question. The old whaling records have full stomachs that have a ton of krill in them, and they clearly can't process that all at once. I think it's a combination of the two things that the fish escape, and even when their mouths are closed and they're expelling water, stuff still will get out. But you're very right. You couldn't fit an entire burrito in your stomach without chewing it. So you need to take bites, and I'm sure there is some assimilation that limits that. I've tried, but it doesn't work. Yes, sir.
Engineering-wise, what would you like to see, if any, improvements in the critter cam map visualization instrumentation?
Yeah, it's a great question. If we had our druthers, what would we make sort of the ultimate tag do? I think honestly having a speaker in there is sort of the most important thing. The tags now also don't geo-reference themselves very accurately, so putting them into space requires us to estimate the speed of travel of the animals, and so we're dead reckoning with those volumes, and being able to put that, make that a bit more accurate, and have these tags, these multi-sensor tags, stay on for longer durations would also be very useful. Instead of a day, have it on for a week at a time. Those are sort of the next level advance. I'm not smart enough to think that far ahead. I only know sort of what's available, but putting an echo sounder in there and being able to, at some level, see what the prey is like and the animals, where the animals are feeding would also be a nice thing. I don't think it wants to carry around a transducer, but Yes.
Well, your burrito example,
blue whales and suggesting that feeding of blue whales at depth, bringing krill and their associated trace element loads back to the surface is responsible for a couple gigatons of primary production in the surface.
Yeah.
And is it possible to test that I mean, here you have a way of looking at where the animal is feeding. Where is it defecating?
That's a great question. It's not a trivial thing. I mean, surface observations are one thing. I'd love to put I mean, half the time we don't put a tag right on the front of the animal. It ends up kind of sliding back on the animal. And you could probably, if you had enough data, go through and look for some sort of acceleration signal in the tag that might indicate when they're defecating. But yeah, you can also rotate the camera on a critter cam. It'd be hard to get it to stick down there, but
Is there an acoustic signal?
not that I'm aware. Manatees, absolutely, but on big whales, don't know. Yeah, I completely buy that biomixing sort of stuff, though. Yeah, it's a good question. Yeah, yeah.
All right, thanks, Dory.
Yeah. All right, thank you, guys.